Hi guys, this is Lydia and I am going to be explaining the differences between the light reaction and the Calvin cycle, which is also known as the light dependent or light reaction cycle. So here we're going to start off at the top with the thylakoids, which is both known as the light independent and light dependent reactions that take place in the stroma. So basically thylakoids are pigment-filled discs which make up the inner compartments. Essentially we're going to start right over here with the photons of light, and basically the whole concept of this is to recharge ADP to ADP and recharge NADPH and generate oxygen and sugars at the end. So basically the thylakoid is full of proteins. They start off with a photoexcitation or photolysis, which is the splitting of water. And then oxygen, protons, and low energy electrons must pass through the stroma before they go to the lumen. Oxygen then fuses out through the stromatic buildup and membrane of the thylakoid. Basically the electrons also, it's really important here. So the first part of the chain, they'll start off with photosynthesis 2. So basically the electron will go down the electron transport chain, which is basically going from high energy to low energy, and it kind of reduces through every step, and it's transported to the plastoquinone, which also delivers electrons to the next protein, which is called the cytochrome, which is also known as a proton pump, which sucks protons out of the stroma into the thylakoid, powerful concentration gradient, and then to the plastocyanine. Basically, the plastocyanine's job is to make sure that photosynthesis 1 has enough electrons. So from photosynthesis 1, you go to the ferredoxin, and then from the ferredoxin, NADP reductase, which is also known as a protein, a major protein over here. And basically that does is recharge NADPH, which is known as a carrier. It's 1 proton to 2 electrons, and recharge ADP. Basically you want to move it back down a gradient called chemosmosis, try to achieve by passing through ADP synthase and achieve equilibrium charge ATP called phosphorylated. Over here is the Calvin cycle, which uses ATP, NADPH, and several other byproducts like CO2, which is very important. Basically, you add all the reagents, which is 6 waters, which comes into the soil, and then it's exited out through the xylem. You start here with the 6 RUBPs, which is also known as ribulose. And then each one has 5 carbons each, so you have a total of 30 carbons, which is very important. And the ending product was the sugars. From there you go to the 6 intermediate, which is adding one of those 6 RUPs with 1 carbon, and then it breaks apart to the 12 PGA, which is also known as phosphoglyceric acid. And basically you just go through here to the 12 PGA. You have to do some rearranging basically, so you go back and forth from the light dependent, charging ATP to ADP and ADP plus to NADH in order to come back here and create the sugars that are needed.